Dr. Sage here. Welcome to the third video in this series where we learn about cell membranes. In the last video, we discussed why something would want to cross a membrane from one side to the other. That's the process of diffusion. Well, in this lecture, we're going to talk about how something can cross a membrane, how it can get from one side to the other. So of note, membranes have selective permeability, which means some things are allowed to cross a membrane and other things are not. And things do need to have the ability to cross a membrane. The cell needs to bring in resources like food or energy. It has to be able to bring in oxygen. And it has to be able to get rid of waste products like carbon dioxide. Now, it turns out that different substances have different abilities to cross the membrane. Some things can very easily go right from one side of the membrane through to the other side. Okay, so the things that can easily cross from one side of the membrane to the other are things that are hydrophobic. So that would be non-polar molecules. So if something is hydrophobic, it will easily cross from one side through to the other side of the membrane. Why is that? Since it's hydrophobic, it doesn't mind passing through the middle of the membrane, which is hydrophobic. So it will easily cross right from one side through to the other side. Now, other things will not as easily cross a membrane from one side to the other side. Those are things that are hydrophilic, which would be polar molecules like glucose or sugar, or ions, which have an electrical charge. So if you have a hydrophilic molecule, like let's say a sodium ion or a glucose molecule, which is a polar molecule, so it has some partial negative and partial positive electrical charges, these things will not easily cross through the membrane. Why? Because since they're hydrophilic, they do not want to go into the center of this membrane that is hydrophobic. They don't want to pass through the center of the membrane. But these things do need to be able to get into and out of the cell. Like you have to be able to bring glucose into your cell. That's the food for your cell. So how does that happen? Well, in that case, the molecule has to pass through some kind of protein to get from one side to the other side. Things that are small and hydrophobic, which means nonpolar molecules, can easily cross from one side of the membrane to the other by going right through the phospholipid bilayer. Things that are hydrophilic, which is polar molecules and ions, cannot easily cross through. They need a protein to pass from one side through to the other side. So, there's different types of diffusion. Remember, diffusion is passive transport. It does not require the cell to spend any energy to make it happen. The analogy I gave you in the last lectures is like water rolling down a hill. You don't have to push water down a hill. Water will roll down a hill all on its own. Okay, no work has to be done to get water to roll down the hill. Well, with passive transport, no work has to be done. One type of passive transport is called simple diffusion. That's when things are going from an area of high concentration to an area of low concentration by passing right through the phospholipid bilayer. That's simple diffusion. The other type of passive transport, so the other type of diffusion, is called facilitated diffusion. This is for things that cannot go right through the phospholipid bilayer. So remember, that it's going to be things that are hydrophilic. These things need a protein to help them cross from one side through to the other side. Now, there are also two different types of proteins that are going to be used. Those are called channel proteins and carrier proteins. So to begin, channel proteins. Basically, a channel protein is a transmembrane protein, so a protein that's embedded in the membrane and spans the membrane, crosses all the way from one side through to the other side. And it has a tunnel or a hole or a channel through the middle. And that allows things to easily go from one side of the membrane through to the other side of the membrane. This is still diffusion, it's still passive transport, which means the net movement of molecules is still from an area of high concentration to an area of low concentration. That's the direction the molecules are going to go. Now, channel proteins, one of the things most commonly used for is ions. So let's say these are sodium ions. Okay, well, that ion that has an electrical charge, it can't pass through the phospholipids, so it can pass through this ion channel protein, which will let it pass from one side through to the other side. Those ion channel proteins are also often gated ion channel proteins, which, just like a gate can be open or can be closed, this channel protein can be open like it's shown here, or it can be closed off and not let things pass through. Okay, so something's going to tell this channel protein that you should open up to allow these ions to pass through. That's going to be a molecule that's going to bind to the channel protein. That's going to be called a ligand or a stimulus molecule. It's a signaling molecule, and it tells the ion channel protein to open up to let the ions pass through from one side through to the other side. Now, we'll learn in more details in later lectures exactly what signaling molecules are. So we'll come back to that concept later. But that's a channel protein. The other type of protein that's used for facilitated diffusion is called a carrier protein. A carrier protein is, again, for things that cannot pass right through the phospholipid bilayer. So again, for hydrophilic molecules. The difference is, while a channel protein basically is just a protein as a hole through the middle, okay, a carrier protein, when the thing that wants to cross from one side to the other, when it binds to the carrier protein actually changes shape to allow that molecule to pass from one side to the other. So here, let's say this is a glucose molecule. Glucose is hydrophilic because it's a polar molecule, so it cannot go right through the phospholipids. So that glucose molecule wants to get from one side to the other. Well, when that glucose binds this carrier protein, it causes that carrier protein to change shape, which then allows the glucose to cross from one side through to the other side. Now, this is still passive transport, so it's still when things are going from an area of high concentration to an area of low concentration. Okay, and it's still passive transport, which means the cell still does not have to spend any energy to make it happen. So passive transport, which is diffusion, requires no energy from the cell because things are going from an area of high concentration to an area of low concentration. Much like water rolling down a hill doesn't require energy to make that happen. It will happen all on its own. But let's say that you want water to roll up a hill. Well, water is not going to roll up the hill on its own. If you want water to go up a hill, you have to use energy, like use electricity, to pump water up the hill. Similarly, if you want molecules to go against their concentration gradient, if you want them to go from low concentration to high concentration, that's going to require energy. Okay, so that's the other type of transport, which is called active transport. Okay, so active transport requires energy because it's making things go against their concentration gradient, making them go from low concentration to high concentration. Okay, so here's an example of active transport. This is a protein that's embedded in the plasma membrane. This protein, what it wants to do is it wants to take sodium ions from a low concentration inside the cell and pump them to an area of high concentration outside the cell. So since you're making it go against its concentration gradient, that's going to require energy. Now, what this protein is going to do is it's going to use the energy in the rechargeable battery ATP. All right, now we're not going to go through all the details of exactly what ATP is and how it works in this chapter. We're going to learn that in the future lecture. So for now, just know that ATP is like the cell's rechargeable battery. Whenever it needs energy, it uses ATP for that energy. Okay, so using the energy stored in ATP, which again we'll learn that detail in a later chapter, what it does is it pumps those sodium ions against their concentration gradient from a low concentration to a high concentration. When this happens, the protein also changes shape, which then allows it to bind to another ion, potassium ions. And when the protein goes back to its normal shape, it then pumps those potassium ions again against their concentration gradient from low to high concentration. Okay, so this is active transport. Why? Because it requires energy to pump things against their concentration gradient. That's active transport. Now, with active transport, there's going to be different proteins that can be used. One type of protein is called a uniporter. Okay, this prefix uni, that means one. So a uniporter pumps one chemical in one direction. That's a uniporter. Then you can have a symporter, which is also called a co-transporter. So two names for the same thing. Symporter also means co-transporter. It takes two different chemicals and pumps them in the same direction. Okay, or you can have an antiporter that takes two different chemicals and it pumps them in opposite directions, like one into the cell and one out of the cell. So the example I just talked about, the sodium-potassium pump, that would be an antiporter. It's pumping sodium out and potassium in. Okay, so those are types of proteins that are going to be used with active transport. Now, when you have active transport, when, for example, you're pumping sodium ions against their chemical gradients, you're making them go from low concentration to high concentration, remember what you're pumping. You're pumping an ion. Well, an ion has an actual electrical charge. Like sodium ions have a positive electrical charge. So what you're doing is you're putting more positive electrical charges on one side of the membrane, and you're making them not have as many positive charges on the other side of the membrane. So by doing this, what happens is you create a lot of positive electrical charges on this side of the membrane. And since there's not a lot of positive charges on this side of the membrane, you end up having an overall negative electrical charge on this side of the membrane. So what that means is you create an actual electrical voltage difference across that membrane. Okay, and there's a term for this that is called membrane potential. So membrane potential is the voltage difference across a membrane, and that's created by these ion pumps that are pumping ions from low concentration to high concentration. Okay, so that's what membrane potential is. When you do this, when you pump ions from low concentration to high concentration, they don't like that. They want to go back across this membrane from this side to the other side. Now, there's actually two reasons they want to go back across that membrane at the same time. One, we've been talking about this whole chapter, which is the concentration difference. There's a lot of sodium ions here, not a lot here. They don't like that, so they want to diffuse from high concentration to low concentration. That's what they want to do, just like everything wants to do that. Okay, so that's a chemical force, the concentration gradient. But there's a second reason at the same time they want to move from one side to the other side, and that's an electrical force. So with electricity, opposites attract. If you have a positive electrical charge and a negative electrical charge, they're drawn towards each other. They want to stick to each other. Okay? But if you have two of the same, like you have two positives, they push away from each other. They repel each other. Two negatives, they push away from each other. Positive and negative are drawn towards each other. Positive and positive push away from each other. It's not the exact same physics property, but it helps you to think about it. It's kind of like magnets. Okay, I'm sure you've played with magnets at some time in your life. Okay, if you're playing with magnets, if you take the north end of one magnet and the south end of another magnet, what happens is they pull towards each other, they stick together. Now take this magnet and flip it around, try to push those two north ends together, and they push away from each other, they repel each other. Same thing happens with electrical charges. Two of the same, like two positives, push away from each other. Well, we just created a bunch of positive charges over here on this side of the membrane. Okay? They don't like that. They don't want to be near each other. They want to push away from each other. So they want to go back to the other side of the membrane for the second reason, an electrical force. There's too many positive charges near each other. So these two together are called the electrochemical gradient. Okay, there's an electrical force, okay, all the positive charges being too close to each other, and a chemical force, a lot of something on one side of the membrane, not a lot on the other. So for those two reasons, these ions want to go from one side to the other. However, since they're an ion, they cannot cross right through the phospholipid bilayer because they're going to be hydrophilic, so they won't go through the hydrophobic center of that membrane. So with active transport, there's a few different types. One type is called primary active transport. Another type is called secondary active transport. Okay, in primary active transport, the protein doing the pumping is the protein that's directly using ATP. So, for example, in that sodium-potassium pump, this protein that is pumping sodium ions is the same protein that's directly using ATP. That's primary active transport. Now, secondary active transport, the protein doing the pumping is indirectly using ATP. Okay, it doesn't directly use ATP. Instead, it uses the electrochemical gradient that's created by primary active transport. So in this primary active transport, we are directly using ATP to pump sodium ions against their electrochemical gradient, creating a high concentration of sodium ions here, a low concentration here. They don't like that, so they want to move back across this membrane. But since they're an ion, they cannot cross right through the phospholipid bilayer to get from one side to the other side. The only way they can cross from one side to the other side is if there's a protein that will let them cross from one side to the other side. And it turns out that there is a protein, this one here. This protein will let them cross from one side through to the other side, but it will only let them do that if they pull this glucose molecule along with them into the cell. So an analogy. Let's say that I am using electricity to pump water up a hill. Now water doesn't want to be pumped up the hill. Water rather be down at the bottom of the hill. But I'm using electricity to pump water up that hill. Now if we're up to the water, as soon as I pump it up the hill, it's just going to roll right back down the hill. But when I pump it up the hill, I put it behind a dam, so it can't roll back down the hill. Okay. Now that dam has a little gate in it that can open to let that water roll down the hill. But I am only going to open that gate to let water roll down the hill if that water takes a little rubber ducky along with it down the hill. Okay, if the rubber ducky is not there, I'm not going to open the gate. Now, so if the rubber ducky is on the water and I open the gate, then when the water rolls down the hill, it will pull that rubber ducky along with it down the hill. Now the rubber ducky wouldn't go down the hill on its own. If it's just sitting on the ground at the top of the hill, it's not going to roll down the hill on its own. The water is going to pull it down the hill. Okay, so in that analogy, the water would be an analogy for sodium ions, and the glucose is the rubber ducky. So the water pulls the rubber ducky down the hill. So this half of the figure, that is secondary active transport. This protein here is doing active transport. It's pumping something against its concentration gradient. It's making glucose go from low concentration out here to high concentration in here. Glucose doesn't want to do that. Okay, but it's not directly using ATP to pull the glucose in. Instead, it's using the electrochemical gradient, all of these sodium ions out here and not many in here. It's using that electrochemical gradient to pull the glucose in. But secondary active transport wouldn't work without primary active transport. This won't work unless you have a bunch of sodium ions out here. The rubber ducky won't go down the hill unless you have water at the top of the hill. So in this figure, this would represent primary active transport, and this would represent secondary active transport. All of the things we've discussed so far is how you get things that are relatively small into and out of the cell. Like ions are very small, glucose is a relatively small molecule. But there are also times you need to get very large objects into and out of the cell. That happens through a process called exocytosis and endocytosis. Okay, so exocytosis is how you get large objects out of the cell. To help me remember this, exo kind of sounds like exit, at least a little bit to me. That's how I remember it at least. Exo, exit, that's how you get big things out of the cell. Okay, so what happens is, inside the cell you have a vesicle, and inside the vesicle is whatever your cell wants to get rid of. It sends that vesicle to its plasma membrane. The membrane of the vesicle then fuses with the plasma membrane, so it becomes the plasma membrane. So now the contents, what's inside the vesicle, is now pushed out of the cell. That's exocytosis, how you get large things out of the cell. The opposite of exocytosis would be endocytosis. That's how you get big things to enter the cell, to come into the cell. How I remember that is, to me, to my mind, endo sounds like enter, kind of. Use your imagination a little bit. Endo, enter. Okay, that's how you get big things into the cell. Now, there are three types of endocytosis. Phagocytosis, which stands for cellular eating. Pinocytosis, which stands for cellular drinking. And then receptor-mediated endocytosis. So the first of them, phagocytosis, we already talked about briefly in the chapter where we learned about the cell. Remember, what cells do is they take a large, let's say, chunk of food that's outside the cell, and then they'll start to wrap their plasma membrane around that piece of food, and they keep wrapping the plasma membrane around that piece of food until it pinches it off, bringing it inside in a vacuole. So this food vacuole, which is also called a phagosome, contains that piece of food. And then remember, in the last chapter we talked about how the lysosome that contains digestive enzymes fuses together with this food vacuole to break apart this food to feed that food to the cell. So phagocytosis is cellular eating. So they take a large solid object and bring it into the cell. That's one type of endocytosis. Another type of endocytosis is called pinocytosis, which stands for cellular drinking. Same concept as phagocytosis. The difference is, instead of one large solid object, it's a bunch of dissolved things, so stuff dissolved in the water. Okay, but again, you pinch in your plasma membrane, and by doing so, you bring in the stuff that's in that water that was outside the cell. So that's pinocytosis, cellular drinking. How I personally remember this is, I think Pinot Noir, which is a type of wine. You might drink wine, well, wine, Pinot Noir, pinocytosis, cellular drinking. That's how I remember it, at least. Third type of endocytosis is called receptor-mediated endocytosis. The main difference with this one is it's very specific. So phagocytosis, you bring in any large chunk of food. Pinocytosis, you bring in any group of dissolved molecules. But receptor-mediated endocytosis is where your cell is looking for something in particular. There's some molecule it needs, and it brings a high concentration of that particular molecule. So, for example, in this figure, this cell just does not have enough yellow stars in its life. It needs more yellow stars. Okay, so what happens is it places these green Ys on its plasma membrane. Those are called receptor proteins. What the receptors do is anytime a yellow star just by chance wanders by, the receptor grabs onto it and holds it. Okay, so that receptor is holding on to that yellow star. And it will only grab onto yellow stars. It won't grab onto blue moons or red circles or orange diamonds. It will only grab onto these yellow stars. Now, since every time a yellow star wanders by, it grabs onto it, then when it brings in its plasma membrane, then it will bring in a high concentration of the thing it's looking for, the yellow stars. Okay, so that's called receptor-mediated endocytosis because its endocytosis is bringing things into the cell mediated by a receptor protein. Remember, these are called receptor proteins. Okay, and it's very specific in what it's bringing into the cell. Okay, so that's our discussion on how things actually enter and exit the cell. And that's the end of our chapter on the cell membrane. So until next time, this has been Dr. Sage.